This is, as I said, this is really one of my favorite chapter. My research, my PhD was on an enzyme. And so I did a lot of enzyme kinetics, studied enzyme mechanisms, designed inhibitors for enzymes, even after my PhD. And that's what we do even today in the lab. So it's a really, fascinating chapter with lots of new and exciting information for you. So yeah, please stay tuned. Enzymes, are biological catalysts, and catalysts, what they do, right? They speed up a reaction. And so somebody had done some preliminary analysis back in 2015, I believe, and they had predicted that there are about 2,700 enzymes in our cells. And they approximately catalyze about 900 biochemical reactions. Virtually most reactions in your cells are catalyzed by enzymes. Okay. And so, but of course, there are some reactions that can also be catalyzed by, I meant proteins, enzymes that are proteins. So, what is a reaction? Reaction is when you take a substrate and convert that to the product, right? And so a catalyst, typically what they do is they will try to speed up the reaction. Okay, so let's say if a reaction of A to B is thermodynamically unfavorable, just a catalyst can do nothing about it. Okay, so if a reaction is thermodynamically favorable, has a free energy negative, an enzyme can actually speed up that reaction. Okay. So we'll talk more about that as we move through this chapter. So we'll learn what are the important features that define an enzyme. Can we actually study enzymes using a mathematical form? So in that context, we will talk about Michaelis-Menten equation that describes enzyme catalyzed reaction. Okay, and that's the main learning, one of your main learning here in this chapter. Okay, so we will sort of not fully derive. We have omitted some algebraic exercise, but we will talk about the equations of Michaelis-Menten and what is its significance and what are the different parameters of Michaelis-Menten kinetics, etc. Then we will briefly talk about the inhibition of enzyme activity, and many times, as I have alluded to you in my earlier lectures, we work with enzymes and we are constantly trying to develop inhibitors of the enzymes that we work on. And our hope is that when we have inhibitors of the enzymes that we are interested in, maybe someday we can create a drug-like molecules. And so in a lot of, in pharmaceutical companies, one of the main area of research is often finding inhibitors, novel inhibitors of certain enzymes that might be involved in certain types of diseases. Enzymes are very specific, so they recognize their substrates in a very specific manner. Of course, there are some enzymes that are somewhat promiscuous as well. And so, but in general, enzymes are known to be specific. So how is specificity built into the enzymes? It's actually built into the active site pocket where the reaction happens. And the last question is, are all enzymes proteins? And for that, the answer is most enzymes are proteins, but there are some enzymes that actually are not proteins. They can be an RNA molecule or a modified DNA molecule. But most enzymes are protein. And is it possible to design an enzyme? Yes, you can design an enzyme and catalyze a desired reaction. And we'll talk briefly about that in the next lecture, which will be on Thursday. Okay, so virtually all reactions in cells are mediated by enzymes. And enzymes typically, what they do is they catalyze thermodynamically favorable reactions. Okay, that is the key, causing them to proceed at an extraordinary rapid rate. So enzymes are the agents of metabolic pathways that provide cells with the ability to exert kinetic control over thermodynamic potentiality. Okay, that's a very loaded sentence there, right? And that is actually referring to something that I said earlier. You know, a thermodynamically favorable reaction may happen at a very, very slow rate, okay? Or may not happen at all. It may require a catalyst, okay? So kinetic controls is what enzymes provide over the reactions that are thermodynamically favorable. Okay, so enzymes can reduce the rate. Depending on some enzymes can get post-translationally modified, and therefore they can have several tiers of activities. Okay, so we'll talk about that as we go through the metabolism class. You will see some fascinating examples of that. Living systems, they use enzymes to accelerate and control the rates of very, very important, biologically important biochemical reactions. All right. So with that brief introduction to enzymes, let's talk about the classifications of enzymes. Okay. So as I said, there are thousands of enzymes but they are broadly classified into 6 of these classes. Okay, this is international classification of enzymes. You can see on the left hand side, they're telling you about the class number, and then they're telling you about the class name and the type of reactions they catalyze. Okay, so oxidoreductases are the redox enzymes, obviously, and they catalyze reactions that involves transfer of electrons, and that could be in the form of hydride ions, right? And then you have transferases. Transferases are enzymes that would transfer certain groups. So they will catalyze group transfer reactions. One of the example of transferases would be the reaction catalyzed by protein kinases that actually transfers the gamma phosphate from ATP to their protein substrates. Okay, so group transfer reactions. Then you have hydrolases that will catalyze hydrolysis reactions, right? For example, proteases, that would be an example of hydrolases. Proteases, what do? Proteases will cleave peptide bond. They will basically use water and hydrolyze that peptide bond to create a free amine and free carboxylic acid. Then you have lyases, isomerases, and ligases. Okay, so some of you, if you're doing any kind of biological research, you may have heard about ligases, right? Ligases are these enzymes that catalyze condensation reactions in which 2 atoms are often joined by a chemical bond using an ATP or any such high energy source. Okay, so you will see when, as we talk about a little bit later on, there are synthetases. Synthetases are these enzymes that we use ATP as the source of their energy, and they are in the ligases family of enzymes. Okay, so one example again will be DNA ligase, and DNA ligase closes, is the enzyme of DNA repair pathways. It basically closes the break in DNA molecules, and usually requires either ATP or NAD+. So it is highly utilized in genetic engineering ligases. Ligases are the enzymes that typically catalyze, cleavages, okay, in which oftentimes electronic rearrangements happen. Okay, so and then you have the fifth class, which is isomerases. Isomerases are the enzymes that will basically transfer a functional group within a molecule from one place to the other. Okay, so you should know, broadly speaking, the international classifications of enzymes. One enzyme that we will talk at length as we go through our metabolism is this pyruvate dehydrogenase complex, PDH complex, which we call it. PDH complex catalyzes, the oxidative cleavage of pyruvate. Okay, so pyruvate will break down to release carbon dioxide. It catalyzes the oxidative cleavage of acetyl coenzyme A to generate pyruvate and carbon dioxide. And so that would be an example of ligases. Now some enzymes require some additional components for their activities, okay? And so in that regard, we want to introduce you to something called cofactors. Okay, cofactors are these additional components that you need for enzymatic activities. And these cofactors are typically molecules or ions that are typically bound very tightly to the enzyme active site, okay, sometimes covalently. So, so if you look here in this table, you'll see that I'm showing you some ions, some cofactors on the left-hand side, and their corresponding enzymes, right? So cytochrome oxidase, which is an enzyme that you will see when we talk about almost the last chapter, oxidative phosphorylation, that actually needs a copper +2 ion. Cytochrome oxidase, catalase, peroxidase, they actually need Fa2 or Fa3+. Now, catalase is a very interesting enzyme that is, commonly found enzyme in all of the living organisms that actually utilizes oxidative metabolism. And it turns out that when we carry out oxidative metabolism, we actually produce trace amount of hydrogen peroxide. And so that hydrogen peroxide must be broken down very rapidly as soon as it is generated to water and oxygen. And catalase does that. Catalase will take a molecule of hydrogen peroxide and will decompose it to water and oxygen. It's one of the highest turnover numbers of all enzymes known. Okay, and I have not defined turnover number yet. That will come into when we talk about Michaelis-Menten kinetics in the next 15-20 minutes. But just to give an idea is that catalase's turnover number is 40 million. Okay, so what does that mean? That means that one molecule of catalase, the enzyme, can convert about 40 million molecules of hydrogen peroxide to water and oxygen each second. Just think about, just marvel the reaction rate here. One molecule of catalase can convert 40 million molecules of hydrogen peroxide to water and oxygen per second. That's the beauty of enzymes. Okay. Catalase, is a tetramer of like 4 polypeptide chains, and each one of those chains has about 500 amino acids or so, and it also contains 4 of those porphyrin groups that we talked about in chapter 5. Pyruvate kinase, again, it's a metabolism enzymes. Many of these enzymes, you'll be seeing them during this lecture. That's beautiful, right? We'll talk about kinases such as hexokinase that requires magnesium +2. Arginase, which when we talk about urea cycle, that's manganese +2. Urease is an enzyme that typically is found in bacteria. Okay, and so that actually will catalyze the hydrolysis of urea into ammonia and CO2. So so that's the cofactors. Then there are coenzymes are the next additional components that you might need for the enzymatic activities of certain enzymes. What are coenzymes? Coenzymes are typically a complex organic molecules that are needed, okay, and can act as a transient carriers of specific functional groups. So what do I mean by that? Remember I showed you the molecular structure of biotin in the earlier class? Yeah, so these are complex organic molecules like, for example, biotin, and they are utilized for doing type of chemistry where a functional group has to be transferred from one place to the other, one protein to other protein. Okay. And so I have put stars here, and all of these coenzymes, you will see them in this class. Okay, when we discuss metabolism and enzyme mechanism, as we go along in this class, you will see that these coenzymes are playing very, very important role, especially when we talk about pyruvate dehydrogenase complex, a beautiful, beautiful machine that actually takes still coenzyme A and converts it to pyruvate and carbon dioxide, that actually uses 5 of these coenzymes. Okay, so when we talk about that, I will remind you that we talked about this briefly. So you have coenzymes that are typically derived from the vitamins that you ingest. That's why people say that eating vitamins is very important from your natural sources, because if you don't have those vitamins, you will not have those coenzymes that are often synthesized using from some of these vitamins. Some coenzymes you have pathways to synthesize your own, but in certain cases you need the dietary precursors. So biotin, biocytin, which is a chemical molecule that forms by if you think about lysine has a side chain, right? And the amino group of the side chain is called epsilon amine. If you read your textbook, and you may have noticed that they refer to that amino group as epsilon amine. So, and biotin itself, if you remember, it has the valeric acid side chain, right? Carboxylic acid. And so epsilon amine of lysine forms an amide bond with the valeric acid side chain of the biotin, and that's what biocytin is. And basically what it does is it transfers CO2 group from one place to the other. Okay, so pyruvate carboxylase, for example, will be an enzyme that will utilize biocytin, okay, to transfer carboxylate group from one substrate to the other. Coenzyme A, if you remember, we talked about this briefly, is a coenzyme that is involved in the transfer of acyl group. Acyl group is nothing but, remember, if you have carboxylic acid, fatty acid, for example, RCOOH, RCO will be considered acyl group, right? Your organic chemistry, you know what acyl group is, I hope. And so coenzyme A is involved in the transfer of acyl group from one protein to the other. And that's the dietary precursor in mammals from which it is synthesized. FAD, called flavin adenine dinucleotide. This is a redox coenzyme that actually transfers electrons, and it's derived from vitamin B2. Lipoic acid. Lipoic acid is synthesized in our cells and is not considered a vitamin. Okay, so there is we have a pathway to synthesize lipoic acid. It's just not very well understood. So you'll see that it's not required in diet. NAD, you will see NAD plus, nicotinamide adenine dinucleotide. This is another redox cofactor that also is involved in the transfer of electrons in the form of hydride. And its dietary precursor is nicotinic acid, and so on. So, so sometimes, you know, students can ask, you already, I think, got a very good idea about the type, the rate at which an enzyme can catalyze a reaction, right? And I gave you an example just to get you a feel for it. The example where catalases can actually break down, you know, 40 million molecules of hydrogen peroxide per second, right? So there, you know, that rate enhancements are tremendous, can be tremendous. So how do you calculate rate enhancements? It's very simple. You basically add enzymes, which you typically add in catalytic amount, right? Sub stoichiometric levels. And basically measure the rate when it is you have not added the enzyme versus when you have added the enzyme. And that's what rate enhancement is, the ratio. And that ratio is listed here, right? As I said, urease in an enzyme that actually is an enzyme that will hydrolyze urea, right, in bacterial species mostly, into ammonia and carbon dioxide. And you can see its rate enhancement is really, really spectacular, right? Cyclophilin, which is actually an enzyme which catalyzes peptidyl prolyl cis to trans isomerase activity. Remember cis trans isomerization of prolines we talked about in our protein class. This is actually peptidyl prolyl cis trans isomerase, cyclophilin, and its rate enhancement is not that dramatic. It's, you know, only 100,000. But so it gives you an idea that, you know, rate enhancement can vary from one enzymes to the other, and in some cases it can be spectacular. Now with this, I want to bring it to this slide, where most of the terms you have learned very well in your general chem classes, okay, general chemistry classes. But still there is tremendous amount of confusion among students about free energy and standard free energy and activation energy barrier. And they know they're all represented by some form of delta G in different places. And so oftentimes students are genuinely confused about them. Okay, so I hope that I will clarify this doubt for you forever. So when we talk about equilibria, right, they are typically linked to standard free energy change, which is this delta G naught, what we call it. And you can see there is a small apostrophe also, people put it, as your textbook has. That's standard free energy change. That reflects to thermodynamics, okay? Reaction rates, when we talk about, they are not linked with thermodynamics. They are linked with kinetics, and they are linked to what we call activation energy barrier, which is often represented by delta G with this double dagger. This is the sign you should learn and use in biochemistry. Okay, so activation energy barrier is this delta G double dagger. That is a kinetic term, and delta G naught is the thermodynamic term, free energy change. Now, free energy change, if you remember from your equation that you learned, is written here in the bottom. Free energy is equal to delta G. So delta G is equal to delta G naught plus RT ln concentration of C times concentration of D divided by concentration of A times concentration of B. I hope you remember that that equation is well known to you, where delta G is the actual free energy of the reaction we are talking about here. What is the reaction we are talking about here? A and B reacting to giving C and D. C and D are product, A and B are reactants. So the actual free energy, what you call delta G, remember what I'm saying, I'm paying attention to here. Actual free energy, real free energy, actual free energy is made of two terms. Standard free energy, which is the free energy in when the reactants and products are in their standard states, like 1 molar concentration, right? And so, and plus RT ln, and then what we call this as the ratio of product divided by reactant. So, a reaction can have delta G naught, which is the standard free energy positive, but can still proceed favorably. Why? Because the sum of these two terms, as long as they are negative, your actual free energy will be negative, and therefore the reaction will be spontaneous. Okay, so this is very important to understand. So many times, if this is positive, first term, which is delta G naught, is positive, as long as RT ln product by reactant is overall negative, more negative than delta G, then your actual free energy will become net negative, and therefore reaction will proceed. How can you make that term more negative? Well, you can make that term more negative if you continue to create a condition in which the concentration of C and D remains smaller and smaller than the concentration of reactants. So if your product C and D are being utilized right away as they are being produced in the next set of reactions, then you can imagine that that can give rise to a negative term. Extremely important, simple concept, but most students are not clear about this. So make sure you know this. Okay, so when there is no driving force, actual driving force, right? Delta G is negative. When a reaction has reached equilibrium, right? Net driving force is 0. So that will be delta G is 0. And so in that case, the equation will change into this equation that is shown here in the note section. Delta G naught, standard free energy, right? Change will be equal to minus RT ln K dash equilibrium. K dash equilibrium is nothing but concentration of C times D divided by A times B. That's where this equation 6.3 comes from, directly from there. That's when the reaction is reached equilibrium. So obviously you can see that if you know the value of K dash equilibrium constant, you can calculate the free energy, right? So they are in this table, they are actually showing you for certain K equilibrium constants, the value of delta G. And you can see some of them are positive and some of them are negative here for some values. Now, just because some of these values are negative doesn't mean that these reactions will happen at a rapid rate. Just because, for example, the bottom one has minus 17.1 kilojoule per mole standard free energy doesn't mean that that reaction will happen at a higher rate than the one that actually is showing delta G minus 5.7, the one on the top. There is no relationship. Okay, so that's the difference. This we are talking about thermodynamics. So what about kinetics? Kinetics is rate, and it is represented by rate constants. Rate constant lowercase k we write, right? And so this is the Arrhenius equation, if you remember from your general chem class again, is equal to A e to the power minus delta G double dagger divided by RT. This is the relationship that exists between the rate constants and the activation free energy barrier. And you can see that it is an exponential relationship, right? Negative exponential. So that is what determines the rate constant. So it usually means that a small change in delta G double dagger activation energy barrier can bring about a large change in rate constant because of this negative exponential relationship. You really have to think about this on your own after this lecture when you are listening to this lecture again, and make sure that you get this very right. Okay, so that's something you might want to keep in mind, because we are going to have that discussion again. Now, when people started developing the model to explain the function of enzymes, Emil Fischer came up with this lock and key model. Okay, and lock and key model suggested, right, that enzymes are like a lock and substrates are like the key, and only one type of key will fit perfectly to that lock and will be able to unlock that lock. Okay, so the assumption of Emil Fischer was that enzymes have already pre-organized active site, and the substrates go there in already fully complementary active site, and then reaction happens. Okay. But you already have seen, right, that for a ligand to be able to bind to a protein, perfect complementarity is not something that is often seen. It's rarely seen, in fact. So you saw that when oxygen molecules binds to the first unit of hemoglobin, the second unit of the hemoglobin undergoes conformational change and allows the second molecule of oxygen to bind with a higher affinity. That affects again third and fourth unit, and so on. So there is always what we call induced fit model, right? We talked about that also when we talked about antibody-antigen interactions in the last class. But it turns out that Emil Fischer was not wrong. I mean, it was not from nowhere. There are some enzymes that actually exist that can validate lock and key model, but very few. One enzyme is dihydrofolate reductase. Okay, dihydrofolate reductase is a small enzyme that actually plays an essential role in the building of your DNA and some other processes as well. Okay, and you might want to know that this was the first enzyme that was targeted for cancer chemotherapy. Okay, so when people were trying to inhibit the function of an enzyme so that cancer can stop growing, dihydrofolate reductase was the enzyme that produced the first drug. Okay, and that first drug was aminoterine. I mean, I'm not sure we will be talking about that, but just to tell you that there is it's an important enzyme. But if you look at this enzyme on the left hand side, clearly you will see that enzyme, contour, they're showing the surface of the enzyme, right? And the two substrates are shown here, one in red and one in yellow. This enzyme uses two substrates, okay? So one substrate in yellow is tetrahydrofolate, and the other substrate is NADP+. It's an analog of NAD+, which is in the red. So if you look here in the unbound, here you can see that tetrahydrofolate is bound, but when it's tetrahydrofolate is bound, you can see that there is a nice binding pocket, if you're looking at my cursor, already pre-organized so that NADP plus can go and bind and do the chemistry. So it is nicely already illustrating. Okay. So this enzyme would be in a case where when tetrahydrofolate is bound, there is already a pre-organized binding site for NADP plus, and therefore in both bound and unbound state, there is hardly any induced fit needed to catalyze the reaction. But scientists, after that model, scientists argued that if an enzyme happens to be completely complementary, when we talk about complementarity, right, we are talking about steric complementarity. So let's say you have a bulge in your substrate, that should go in a groove on the enzyme because it should fit, right? If there is a positive charge on your substrate, there should be a negative charge in the enzyme active site. That's what complementarity means, both steric complementarity as well as electronic complementarity. And so scientists believe that if an enzyme was completely complementary to its substrate, then it would be a very, very poor enzyme. And so lock and key model of Emil Fischer, which was proposed in 1894, could be misleading. So why would that be? Why did scientists think like that? To explain that, this is a very fascinating enzyme that I love talking about to my students, and students love it too. Now, this is an imaginary enzyme. Okay, so let's say you have to catalyze a reaction, hypothetical reaction. And that hypothetical reaction is you take a substrate, which is a metal stick, and you want to break that metal stick into product, which is two broken sticks from the middle. So if you do not have an enzyme, obviously you will take two ends of the metal stick and try to bend it, right? So your transition state of that reaction might look like this, where on the other side, on the top face of this metal stick, there will be some metallic bonds that are broken. Now, if you were to draw the free energy diagram for that reaction, you will see that they are showing you on the y-axis free energy, right? Free energy, and x-axis is reaction coordinate. Reaction coordinate is how far the reaction goes. S substrate starts from the left and product is on the right. So you can see that substrate has to go through activation energy barrier. That's the transition state, right? Shown there by double dagger, same. And from double dagger, then basically more and more things break, and eventually you have two metal sticks broken. That's your product. So to catalyze that reaction, you have to go through that transition state, which is this bent stick. But so you have to supply energy, right? So delta G double dagger is that much. That's your activation energy barrier that you need. That much energy you have to supply to bend it. So you can reach the transition state, and at which point you have a shot at getting to the product side. Cool. This is activation energy barrier. This is kinetics we are talking about. So if you really want to talk about thermodynamics here on the same diagram, it will be the difference of free energy between where the S is and where the P is. Okay. So the difference between free energy between S and P would be the thermodynamic delta G, free energy change for that reaction. But delta G dagger here shown is the kinetic one. Cool. So let's imagine a scenario where you do have an enzyme like Emil Fischer's enzyme. Emil Fischer proposed that enzyme is completely complementary to the substrate. That's what she proposed, like lock and key. And so if that's the case, what will happen is that when you form the enzyme substrate complex, where basically enzyme has come closer to the substrate, it has brought substrate into its active site, forming what we call Michaelis-Menten complex. It's a complex which is just non-covalent in nature. It's just a physical attraction between enzyme and substrates based on those 4 non-covalent interactions that we have been talking about. In that Michaelis complex, ES complex, you can see that there are complementary interactions. Sterically, the stick is able to bind nicely, and with red dash dots, they are showing you that they are interacting with the metal stick, maybe electronically. So complete complementarity will mean that you basically, you form the enzyme substrate complex, but it is a dead end complex. You are not able to form product. You are not able to do chemistry because the substrate is completely complementary to enzyme. It has nice binding energy. That is what is shown here in the right hand side in the middle curve, middle plot. Same scenario, free energy, right? So you can see substrate binds, but because substrate is completely complementary to enzymes, enzyme substrate that you form is further stabilized. Its free energy is lowered, right? So free energy is lowered. So ES is, free energy is lowered. But then to be able to go to product, it still has to go through the same transition state, right? So now from going from ES complex to the transition state, delta G of catalysis shown here in blue has increased even more than the one that where there was no enzymes. The one that was no enzyme was one shown here on the top, delta G. Look at the magnitude of that. But when the enzyme is totally complementary to substrate, enzyme substrate complex becomes even lower in free energy, and therefore the activation energy barrier becomes even more high. And so that's what they are showing you here. So that's a bad scenario for catalysis. This is not catalysis. If anything, it's inhibition. Not speeding up the reaction. It will slow down because now your activation energy barrier, as you are seeing in the blue, in the middle, is higher, right? Now, what if enzyme is not complementary to substrate, but is complementary to transition state? Okay, transition state theory. This is the latest and explains most of the thing. Enzyme catalyzed reaction. The item number C. What happens is that enzyme binds to the substrate with somewhat weaker affinity. Not all interactions are utilized by the enzyme active site pocket and substrate. It binds, just about binds. It brings in, say people say, get your foot in the door. That's what is happening for the substrate here. But as binding happens, reaction starts to proceed in the direction of product. And so it goes through the transition state. As it is going through the transition state, enzyme is also undergoing induced fit so that it actually now interacts more strongly with the transition state. It stabilizes the transition state more. You can see here. This figure says a lot about everything that you know about catalysis. Okay, that's why it's so cool. So why? Because remember, in scenario C, enzyme is complementary to the transition state. That's why when you are in the transition state, it is now fully complementary. The interactions are maximally utilized. And therefore, your stick can be easily broken into the two pieces. How? That is what is shown here in the bottom right graph. Substrate here, blue line, pay your attention to blue line, okay? Substrate here forms an enzyme substrate complex, but this enzyme substrate complex is not too stable. Free energy of enzyme substrate complex is not lowered, but somewhere around the substrate. Once you forms the enzyme substrate complex, reaction goes through the transition state. The transition state is complementary to the enzyme, and therefore those interactions will stabilize the transition state. So you can see the black, the plot that is shown here in the dashed black dots. You can see that transition state double dagger barrier is now lowered to the blue double dagger, right? And so in that case, your delta G of catalysis is lower than the uncatalyzed reaction. And therefore, you are able to catalyze the reaction at a much faster rate because transition state is being stabilized. You are lowering the activation energy barrier of the difficult step, the rate limiting step of the reaction. So the induced fit model, where induced fit happens as substrate changes into the transition states. The transition state is the one that actually preferentially stabilized by the enzyme that leads to the enhanced reaction rate. Cool. So that's, people, you may ask question. I understand, Dr. Pathak, you are saying that, transition state is stabilized. That's why free energy barrier is lowered, right? Activation energy is lowered, as you are seeing in the blue lines. But where does that energy comes from? How is it lowered? How is activation energy lowered for the transition state? I have already been alluding that to you, right? Activation energy barrier is lowered, and that comes from the binding energy, delta G binding. That binding energy is the preferential binding energy of the transition state to the enzyme. Okay, and where does that binding energy comes from? It comes from those 4 non-covalent, weak non-covalent interactions that we have been talking about. Electrostatic interactions, right? Hydrogen bonding, hydrophobic, and so on. So that's where, that's how enzymes do catalysis. Now, enzymes also use some other tricks for catalysis. Okay, and one example is that rate enhancement can happen also by entropic reduction, by pre-organizing the substrates into the active site, right? If 2 substrates are just moving about in solutions, the possibility of them reacting is lower than when they are brought together in the enzyme active site, right? They are pre-organized, entropic reduction. And that is what is shown here, okay? They are showing you a bimolecular reaction of a carboxylate group with an ester to generate anhydride. And so they are measuring the rate constant here. This is a second order rate constant, right? So per molar per second. Let's say whatever that rate constant is, you don't normalize that to 1. Now what happens if you can pre-organize the reacting partners by bringing the 2 functional groups in the same molecule as shown here in the B? If you do that, you have somewhat restricted, you have reduced the entropy of the reacting partners by bringing them into one molecule. So you still have single bonds, 3 single bonds that can rotate freely, but it's within one molecule, these groups are. So if you actually let this molecule react, in this case, because it's a unimolecular reaction, it's a single first order rate constant. That's why the unit second inverse. And if you measure that rate enhancement, that will be, let's say, 10 to the power 5 molar. Okay. So, but if you can further reduce so that these 3 degrees of rotations can be completely eliminated by actually engineering these functional groups in a system like this, right? Cyclic system, bicyclic system, as you see, where these 2 reacting groups are shown in the equatorial position and they are well positioned to react, right? You can see that the bond rotation will not be possible. All those 3 bonds are now restricted. And if you actually look at the rate enhancement, you can see it's 10 to the power 8 molar. Okay. So, you can see that entropy reduction can have significant effect in rate enhancement. Now, you might think that, this one has no unit, this one has the unit of molar and molar, and they have to do that so that they can directly compare, because the top one is a bimolecular reaction and the bottom two ones are unimolecular reactions. Okay. So 8 molar typically, when you see the rate enhancement of 10 to the power 5 molar here, right? It usually means that if the reactants, which would be here, the reacting functional groups, right, were present at 1 molar concentration, right, then these two reacting groups would almost behave in such a way as if they were present at a concentration of 10 to the power 5 molar. Okay, that's what it means, but don't want you to get bogged down with too much. But what you can see that rate can be enhanced dramatically if you can actually reduce the entropy of the reacting partners. Now, what about other assistants? Okay, what would that be? Let's look at a reaction that you are very familiar with so far. Although we have not discussed enzyme mechanism class yet, but you know that peptide hydrolysis, right? You are familiar with the peptide bond. So that's what is shown here. Imagine that this is an alcohol as shown here, but imagine it could be just water. So you can have HOH if you want. So hydrolysis of peptide bond, right, or reaction of an alcohol to an amide, right, to generate an ester and amine as product, whatever you want to look at it. If this reaction has to happen, what has to happen? This nucleophilic oxygen, the lone pair, will attack carbonyl, electrophilic carbonyl. You will push that double bond to generate O minus, that tetrahedral intermediate, 4 bonds to that single bond. But look what's going on here. You are generating extra charges on this intermediate, right? And so if there were no means to actually stabilize this species that you are generating, then eventually you will form this transition state and it will fall back to give you reactant species. You will hardly have any reactions going forward. But if you can provide either general acid base catalysis or specific acid base catalysis, if there are catalysts present, which can be a proton donor, right, or proton acceptor, then the same reactions can be catalyzed at a much appreciable rate. Why? Because look what has to happen to get to the product here, right? Look at here in the middle. What has to happen is that O minus has to collapse to give carbon oxygen double bond, and that NH with R4 has to leave. That is the leaving group. But if it has to leave without any assistance, without a proton or assistance from a proton, then it will be a strong base, NH minus, R4 NH minus. R4 NH minus would be a very, very strong base. And therefore, it will be a terrible leaving group. Reaction will go nowhere. But if you were to provide acid, right, either general acid shown here in the form of HA, or specific acid shown here in the form of hydronium ion in the blue on the left, then what can happen is that you can protonate that leaving group to generate this species at the bottom, R4 NH2 plus, bound to the carbon with a single bond. That now becomes a good leaving group because it's a weak base. R4, it will leave as a R4 NH2 instead of R4 NH minus. And so by providing general acid assistance, you can actually imagine that now you will be able to form the product more readily. Okay, so if you have a general base, which is B with the lone pair they are showing you, or a general acid, which is HA, that's how typically they replace. R specific acids as shown here in form of H3O plus left hand side and OH minus. You can pick up that proton and basically catalyze the proton transfer reaction much more readily, and therefore you have a shot at getting to the product. And general acid base catalysis happens in the many organic reactions, and of course they are prevalent in biochemistry. So how are they prevalent? Who participates? It turns out that the side chains of many of these amino acid residues listed here can actually participate as general acid or general base, depending on which form they exist in the enzyme active site. Remember, pK can be dramatically altered in the enzyme active site. pK of a group depends heavily on its environment. Remember, we already give several examples of that, right? We talked about a comparison of the carboxylate pK when the carboxylate is alpha carboxylate of an amino acid, or when we compared this to acetic acid, for example, right? And we were able to see that pK of the carboxylate changes depending on the environment. So in enzyme active site also it does. So glutamate and aspartate, they have acidic side chain. If they have to act as a general acid, then they have to have their side chain in the form of COH, protonated form. If they have to act as a general base, which means they have to act in such a way they have to pick up a proton to speed up the reaction, then they will be acting as a general base, and general base form will be carboxylate form, deprotonated form. Same goes for arginine and lysines, protonated form as a general acid, deprotonated form, upside chain, general base, and so on. So make sure you write it down and practice it nicely so you are fully aware of this. Now, we are running a little slow. It's only 15 slides we did, and it's already, so we have to speed up a little bit. Now, enzyme activity depends on the pH as well, right? I gave you an example when we talked about proteins in the last class. I'm showing you again pepsin, right? Pepsin is a enzyme, and it is usually stored as a proenzyme in the form of pepsinogen, and it's usually released by the chief cells in the stomach wall. And it's a very, very important enzyme for digesting your proteins, okay? It acts as its best pH, optimal pH is very acidic, around 1.8 or so, okay, near 2. So on the y-axis, you are basically saying log of V0. V0 is the initial rate of the enzyme catalyzed reaction. And on the x-axis, you are measuring that rate at different pH value, and you are plotting it. And you can see that the rate is maximum for pepsin at around 1.5. For glucose 6-phosphate, optimum pH is around 8. It works best at pH 8. Okay. Glucose 6-phosphate phosphatase is an enzyme that we will talk about that. It is actually a very important enzyme of your liver cells, and it helps maintain a glucose level in the blood between your meals. Okay. So pH optimums can be different, right, for different enzymes. But look at this. In a closely packed environment of a protein, the pK of a side chain can be significantly altered. I am re-emphasizing this again and again and again and again because this plays a very important role in your understanding of enzyme function. An enzyme called acetoacetate decarboxylase, okay? When we talk about fatty acid metabolism, you will see this enzyme. This is an enzyme that actually has a lysine group in its active site. Do you know the pK of that lysine in the acetoacetate decarboxylase? It's 6.6. If you remember, the lysine pK is in the free state of lysine is 10.5. So it's 4 units more acidic in acetoacetate decarboxylase, right? And so that lysine chain can act as an acid or base. Maybe that's why its pK is altered so dramatically. So how is the pK of this lysine is so low compared to free lysine? Almost it's 10,000 fold more acidic than the free lysine. Is because it turns out that if you looked into the active site of this enzyme, you will see that there is another lysine nearby. And so if you have two lysines nearby in the active site, one lysine will lower the pK of the other lysine. Why? Because if it lowers the pK of the other lysine, then the enzyme will exist in a form where one lysine will be protonated and the other one will not be protonated. And that will be a happy, more stable um, um, scenario for that enzyme, right? Having two positively charged groups nearby is not going to be very favorable. So if this enzyme evolved to generate this lysine, active site lysine, whose pK is 6.6, all it had to do during evolution is that keep switching one amino acid with the another one until you find that lysine is actually best and the activity of this enzyme is perfect. Very, very cool. You need to think about this a lot more than what I just said. You have to write the acid-base equilibrium of the two lysine and see why that could be. Having another lysine could lower the pK of the other lysine. Let's quickly switch to Michaelis-Menten kinetics now. I have eliminated most of the derivations for Michaelis-Menten equations. And it's relatively straightforward, and you can actually follow that. That's your homework. You follow that in your text and come back and ask me questions during the office hours. Don't ignore it. It's fun. You like it. It turns out that the enzyme catalyzed reactions are measured by rates, initial rates. Okay, so whenever you are measuring an enzyme catalyzed reaction, you always measure initial rate. How do you measure initial rate? How much product you are forming per unit time, how much substrate is being consumed per unit time, right? So you can actually follow the enzyme catalyzed reaction either by following product, increasing product concentration, or depleting substrate concentration. In this plot on the y-axis, you see they are monitoring the product. So that's why product increases with time. And so simple reaction is S going to P with the help of enzyme. So if the concentration of S is 0.2 micromolar, you will see that product forms in the beginning. When all substrate has been consumed to product, there is no net product formation. The curve flattens out with time. This is what we call progress curve. The reason I want to warn you here is that plots look the same. If you don't pay attention to the axis, then you will get thoroughly confused. So that's why very, very important to pay attention to what is the axis, what the two axes represent. This is called progress curve. We are monitoring simply the progress of a reaction with time on the x-axis. So if you increase the substrate concentration to more than 2 times 0.5 micromolar, you can see the rate of the reaction was double, right? And eventually when all substrate was consumed, again it flattens out and so on. So how do you measure the rate? What you do is you basically measure this tangent to when the time is 0. Okay, so you extrapolate, you basically measure the initial rate. Initial rate will be first initial phase, what is the rate? Which means you basically draw a tangent that extrapolates to 0 time and you measure the slope of those. That becomes your initial rate. That's how scientists measure initial rates for an enzyme catalyzed reaction. Remember that. Slope that extrapolates to 0 when you are following the rate of the reaction, when you are monitoring the progress curve. So you see that there are 3 different rates as represented by the dashed lines at 3 different substrate concentration. It turns out that enzyme kinetics is different. Okay, if you had a simple first order reaction, right, your reaction rate will be versus the reactant concentration will be a linear straight line, right? So, but it turns out that enzyme catalyzed reactions are a little more complicated. Very, very important to pay attention. At low substrate concentrations, the rate is actually proportional to the S. So there is a real linear relationship. Rate is proportional to the substrate concentration, and therefore it is in the first order with respect to the substrate. But if you increase the concentration of substrate many-fold, then the enzyme reaction approaches zero order kinetics, no longer dependent on the substrate concentration. So at much, much higher concentrations, how much higher concentrations, I will tell you in a little bit. It follows zero order kinetics, which means it saturates. This is what we call saturation kinetics of enzyme catalyzed reaction. So how does it look like? Saturation kinetics looks like this, as shown here. V, initial rate you are measuring. Okay, initial rate on the y-axis and the substrate concentration on x-axis. This is very, very different than what is this plot. That's why I'm saying this is a progress curve which is utilized to measure initial rate, which will be V0. V0 for one substrate, V0 for second substrate, V0 for three substrates. You have three initial rates you have measured here in this plot. And what you have done is then you have taken those three initial rates and you have plotted against the substrate concentration. That's what this plot says. V, initial rate versus substrate concentration. That's what you see. When the substrate concentration is low, you can see that there is a linear dependence of the substrate. Almost. It's not perfect linear. It's a rectangular hyperbola shape. But it is somewhat linear dependence. But when the substrate concentration approaches higher, as you see, as you go from left to right, you see that the rate, initial rate V doesn't change anymore. Very little change. It will never reach the, V max, what we call it. So when the rate doesn't change anymore at very, very high substrate concentrations, we call that as a V max. That's what capital V max is shown. V max is a theoretical value that you will reach when the substrate concentration is way, way high. How much high? I will tell you in a bit. But that's what it means. Linear first, but then zero order kinetics when the substrate concentration is high. With that, to understand this further, let's derive the Michaelis-Menten equation. Okay. Michaelis-Menten equation was derived by Michaelis, Louis Michaelis and Maud Menten in 1913. Before they derived the equation, they basically assumed certain things. So these assumptions are very, very important. What are the assumptions? Basically, it assumes like this. You have E plus S, enzyme plus substrate, going to form Michaelis complex ES with forward rate constant of K1. K minus 1 is the breakdown of ES to give E plus S. And that Michaelis complex goes to give you free enzyme plus product. Okay, free enzyme goes back and takes another substrate and keep on doing this catalysis, catalytic cycle. Now ES going to E and P is represented by K2. That's the rate constant. And K minus 2 is going from E plus P going to ES. So what are the assumptions? It assumes that you form the enzyme substrate complex. And that is enzyme substrate complex is in rapid equilibrium with the free enzyme. Okay, so that is a rapid equilibrium. And that rapid equilibrium is achieved when your substrate concentration is much, much higher than the enzyme concentration. Okay. And so when steady state is achieved, this is steady state equation. Okay. When steady state is achieved, the concentration of Michaelis complex remains constant. That's what is here. ES remains constant when substrate concentration is way, way higher than enzyme, which means substrate concentration was 1 micromolar. Enzyme concentration should ideally be 1 nanomolar or even less. Then it is easy to achieve steady state. All right. And so the other assumptions they had was that the breakdown of ES to E plus P is the slower step, the rate limiting step. And going back, taking product, going back to form the Michaelis complex, that reaction doesn't exist. So K minus 2 is 0. It's also known as Briggs-Halden's assumption. So K minus 2 is 0. So literally the assumption is that in steady state, the concentration of ES remains constant. Whatever you form in the first step, that's how much you break in the product step. Okay. So with those assumptions, K minus 2 is 0. So you can just remove that. So you just are talking about K1, K minus 1, and K2. K2 is the rate limiting step, slowest step in the reaction. So you know that if you have a multi-step reaction, rate of that reaction is governed by the slowest step, right? So that will be V0 will be proportional to the concentration of ES, because ES to E plus P is the slowest step, the breakdown. So that rate constant is K2. So the V0 is equal to K2 times ES. So you at this time, if you go back and look in your text, you can see that all they have done is that ES is, Michaelis complex. You don't know experimentally the concentration of ES, right? You know what your concentration of enzyme is. You know the concentration of substrate, but you don't know concentration of ES, right? So all they have done is they have actually done some algebraic manipulations to solve the value of ES in the form of enzyme concentration or substrate concentration and plug that value in. And when they do that, they actually get this Michaelis-Menten equation, which is initial rate V0 is equal to K2, and the ES value is right here, ET, which is total concentration of enzymes times substrate concentration, divided by Km. They have introduced a new constant called Michaelis constant, plus S. What is Km? Km is made up of these rate constants. Here I have defined Km. Km is K minus 1 plus K2 divided by Km1. So what is Km? Km is a mixture of different rate constants. In this case, very simple scenario, it is K minus 1 plus K2 divided by whole thing divided by K1. So that's what Michaelis-Menten constant is, Km. And then they define another parameter called Vmax. Vmax is nothing but K2, which is the rate constant for the slowest rate limiting steps, times the total concentration of enzyme. So Vmax is how you define K2 times ET. This right here and the equation you should know very well. Okay, if you don't know the derivations, fine for this class. But the value of these parameters are very important. K2, because it is rate constant for the slowest step, it is also known as Kcat. Sometimes people refer to that. So K2 is a Kcat here, and I'll show you that. So you should know what is Km, what is Vmax, and what is Kcat, etc. And what is Km made up of, what is Vmax made up of. When that you already know, then if you plotted your V0 initial rate versus the substrate concentration, you can see that you are seeing a rectangular hyperbola, right? And you know what I'm just showing you that same thing again. So V0 Michaelis-Menten equation is K2, which could be Kcat, K2 times total enzyme concentrations times substrate concentrations divided by Km plus S. What is Km? Km is K minus 1 plus K2 divided by K1. And Vmax is nothing but K2 times ET. So sometimes Michaelis-Menten people write like this. They will just write V naught is equal to V max times substrate concentrations divided by Km plus S. People write that way. Now let's see how at low substrate concentration rate is proportional to the single concentration of S. Single unit directly proportional to the S. So when you have lower concentration of S, right? Concentration of substrate is low. Then what will happen? Think about this equation. You can just directly see when the S is really low compared to Km value. Okay. Substrate concentration, this unit is low than Km, then you can ignore. It's like, let's say you have 1 and you are adding 0.1. So it will be 1.1, right? The division will be by 1.1, not 1. But the value will not change much. So you can just ignore S here, right? In the bottom, because you are adding S to Km. If S is very, very tiny quantity compared to Km, you can ignore it. If you do that, you will see that your V naught will become V max times S divided by Km, exactly what they're showing you here. And if you look here, you can see that V max is a constant, Km is a constant, V naught is linear with respect to S. Linear dependence of initial rate on S, right here. That's what is shown here. What happens when you have the case where concentration of S is much, much larger than the Km value? When that's the case, you will have to ignore Km, right? And so then what will happen is V naught will be equal to V max times S divided by S, and S will get cancelled. And so V naught becomes your V max. And that's exactly what V max is. No dependence on S. When the substrate concentration is much higher than the Km, then your initial rate becomes V max. That's what this means. That's where you have zero order in S. So typically students ask how much more and how much less. And I always tell students, Km, let's say, is 5 millimolar, right? And if you are using a substrate concentration 1 millimolar, then you are operating under S concentration much lower than the Km, at least 5-fold, right? If Km is 5 millimolar, and if you are using 25 millimolar of substrate concentrations, then you are operating under Vmax conditions, where rate will not depend on the concentration of substrate because you have achieved Vmax status. Pretty cool stuff. Very, very cool. Very easy to confuse. I used to give a break on this, but I will not in this case, but just solve this for you. For enzymes in which the rate limiting step is the following, in the following reaction scheme is the one with K2 as the rate constant. So again, just like the way scenario we discussed, same equation, Km becomes equivalent to what? And they give you choices. Remember what is Km? How is Km defined? I can show you. Km is defined as K minus 1 plus K2 divided by K1, right? That's how Km is defined. But if K2 is the rate limiting step, right, which means smaller, right? So K2, again, you are adding a small number to a large number. So in that case, you can ignore K2, right? K2 is the rate limiting step, smallest rate constant. So then Km becomes K minus 1 divided by K1. And that, you remember, is dissociation constant, if you remember from our earlier discussions. And so the Km becomes the Kd, dissociation constant, Kd value for the ES complex. What is Kd? Kd is the dissociation constant. We talked about that. ES going to E plus S, right? So that's something that you might want to know. Now, Michaelis-Menten kinetics is when the steady state has been assumed, right? Steady state kinetics, it's called. Under steady state conditions, you know that the concentration of ES doesn't change, which means the rate it forms is the same rate it breaks down when the steady state is achieved. So if you were to follow the concentration on the y-axis of different species along with time, you will see that when steady state is achieved, right after this box, the concentration of ES remains constant throughout. That's what the steady state assumption means. And then you are actually see that blue line, you are forming more and more product as the substrate concentration goes down, as the more and more of substrate is being consumed to the product. But during the entire time, the ES concentration remains constant. That's what you have to pay attention to. Even when the substrate concentration is low and you have formed most of the product, the concentration of ES remains constant. That's steady state. But if you had some means to actually discuss, so to look at this initial plot and zoom it out, we call that pre-steady state. Okay. And so in pre-steady states, if you zoomed in, you will see that concentration of enzyme depletes initially, and that happens because you are forming more and more of ES in the pre-steady state, and eventually when the steady state is achieved, you have formed your ES complex, and your concentration of ES complex remains constant, and therefore the enzyme also remains constant as you form more and more product. So this is called pre-steady states. And people do pre-steady state kinetics sometimes to figure out reaction mechanisms and so on. What is Km then? Again, Km is called Michaelis constant. It's a constant made up from the bunch of rate constants. In the simple case scenario that I showed you, where you have only two steps, E plus S going to Michaelis complex EX with the forward rate constant of K1, reverse constant rate constant of K minus 1, and with the rate limit step of K2, then Km is made up of K minus 1 plus K2 divided by K1. That's what Km is. So Km is an estimate. It's not actual, but it's an estimate of the dissociation constant of enzyme and substrate. It becomes Kd when K2 is the absolute rate limiting step. It becomes K minus 1 by K1. But even under these conditions, it gives you a pretty good idea of dissociation constant. It's an estimate. So the Km is like Kd. The lower the value of Km, the tighter the affinity for that substrate is for the enzyme. Just like Kd, lower the value of Kd, the tighter the affinity of that ligand is to the protein. Same thing for Km. Smaller Km means tight substrate binding, higher Km means higher substrate binding. What about Vmax? Vmax is also a constant, right? It's made up of two factors: enzyme concentration, which is the total concentration of enzyme, that is what you have used, and Kcat, which is sometimes can be K2, as I said. But Kcat, if you want to define, you can divide Vmax by Et, that becomes Kcat, right? And Kcat is known as what we call turnover number. Remember, I was telling you 40 million in the case of catalase per second. That is Kcat value. So Kcat is nothing but number of chemical conversions of substrate molecules per second per mole of enzyme. This is why I tell you that for you, it's very important to write down and practice and know the relationships between them. If you don't, then you will not know. Okay. So you know that you can determine Kcat if you know the Vmax and you know the enzyme concentration, right? All you do is take the Vmax, divide that by enzyme concentration, you can get Kcat. And if you do that, you will see that Kcat unit comes out to be in time inverse. So once you know the Kcat, you can also determine Kcat by Km. People do that. That's another parameter. Kcat by Km refers to what we call specificity constant or catalytic efficiency of the enzyme. Okay. And it's a second order rate constant because, you know, the unit of Km is concentration, right? and unit of kcat, as I told you, is time inverse. So it will be concentration inverse time inverse. That's why it's a second order rate constant. And it basically tells you how enzyme performs when the substrate concentration is low. And the upper limit of this is typically the diffusion limit, which is the rate constant, rate at which the enzyme and substrate diffuse together. So with that definitions, let's look at some value, okay? Before we look at the value, it turns out that in earlier days, when people had to do this, the Michaelis-Menten kinetics to determine the Michaelis-Menten parameters. What are Michaelis-Menten parameters? The value of Km, the value of Kcat or Vmax. Then people had to actually do this, measure the initial rate at different substrate concentration and plot it, and try to either do the data fitting, non-linear data fitting that can give you the value of Vmax and Km. Okay. One thing I missed out here, as you can see already here, is that you can actually, from this plot, you can determine experimentally the Km value. How do you do that? What you do is when you did this plot, you already know where your Vmax is, right? Which is right here. So what is half of that Vmax? Let's say here it was 10. Half of that is 5 here. You draw a parallel line. Remember, that's how we estimated KD value in the previous chapter. Same concept. You draw that parallel to the x-axis, see where it cuts the curve, and then you extrapolate that to substrate axis, and that reading is your Km value. Okay. So what is Km value? Km value is the substrate concentration at which initial rate is half of Vmax. And you can actually literally do that substitution and see that for yourself, how that matters. So this is how experimentally you determine Km value. But if you do not want to do this non-linear fit, if you just want students to calculate Km and Kcat and Vmax, etc., you can do what we call linear plots that are easy to do, also known as Line-Weaver-Burk plot, double reciprocal plot it's called, okay, also known as. All you do is you take the Michaelis-Menten equation as shown here. V naught is equal to Vmax S by Km plus S. Sometimes people write V naught, or sometimes they write lowercase of V, which basically reflects initial rates. Just you should know that. And so all you do is you take the reciprocal and rearrange, and then you will get to this equation. This equation is now in the form of y is equal to mx plus c. And therefore, you can plot 1 by y and 1 by s, right? So if you plotted 1 by v naught versus 1 by s naught, as shown here, you will get a linear line in this case. Why? Because you can see y is equal to mx plus c. It's a straight line equation. So if you plotted 1 by v and 1 by s, that's x, the slope of that line will give you the value of Km by Vmax, and the intercept will give you 1 by Vmax. So if you did the experiment, you can see that where your intercept is, that can give you the value of 1 by Vmax. Therefore, you can know the value of Vmax, right? And slope will give you the value of Km by Vmax. So you measure the slope, you already know the Vmax, and therefore you can get the value of Km as well from this, right? Km can also be gotten from this way. Km actually turns out that it will extrapolate on the other side of x-axis at -1 by Km value. So using the double reciprocal plot, you can actually determine Km and Kcat or Vmax value. Cool. So this was much easier for students to just use the ruler, and then just they were given, let's say, if I give you V naught at different S concentration, right? Initial rates at different S concentrations. All you will have to do is you actually take the reciprocal of both of them and then plot 1 by V naught at the y-axis and 1 by S on the x-axis, and then you can get, see where it cuts to the y-axis. That's your 1 by V max. 1 by V max, you can go know the V max, and then once you know V max, you can go get the Km value from the slope. So that's what people did. It turns out that some examples I'm showing you here of Km and Kcat value, okay? Hexokinase is a kinase that we'll talk about in glycolysis. It has a substrate, two substrate, it's ATP and D-glucose. So they are telling you the Km value for that. This is all experimentally determined from the experiments that I just described to you, how you can get Km and Kcat value, etc. You can see here for hexokinase, Km value is, for D-glucose, it's 0.05 millimolar. D-fructose, it's 1.5 millimolar. So you can notice here that D-glucose seems to be a preferred substrate because it binds better to the hexokinase. Km value is much lower than the D-fructose value. Catalase here, same thing, 40, Kcat value is 40 million, right? Its substrate is hydrogen peroxide. Once you know the Km value, you know the Kcat value, all you have to do is take the ratio, and then you can actually know what is the second order rate constant, specificity constant for this enzyme for different substrates. And that's what is listed here. It turns out that acetylcholinesterases are very important enzymes that usually will work to degrade acetylcholine, which is a neurotransmitter. Okay, and neurotransmitters are the way a nerve cell communicates with another cells, like muscle cells, for example. So once the signal is, once the job is done, muscles have contracted, you must destroy the acetylcholine, and therefore this acetylcholinesterase enzyme is crucial. If not, your muscles will keep on twitching. Okay. So it's a very, very important enzyme, and you can see Kcat by Km value is 1.6 in 10 to the power 8 per molar per second, which is almost near to the diffusion controlled limit of 10 to the power 8 per molar per second. So if I were you, actually, I will look at this and stare at this and actually try to get a feel for these numbers. Don't memorize this, nothing to memorize here. With that, I want to now focus a couple of slides on inhibition. I already told you, right, that people have strong interest in developing inhibitors of enzymes. And that's because many of the enzymes activities are dysregulated in disease states. Okay. So for example, there is a cancer growing. We work on a kinase called NEC2 kinase. We know that NEC2 kinase causes cancer somehow. So we want to develop inhibitors so we can block the activity of this kinase, right? So we go and synthesize molecules, and then we try to do kinetics to figure out what kind of inhibitor this molecule is. What is the potency of this inhibitor? So we are talking about here reversible enzyme inhibitors. What does that mean? Reversible enzyme inhibitors are that when you throw enzyme inhibitor in the mixture with the enzyme, inhibitor will go and bind to the enzyme. And if you want to get rid of that inhibitor, all you have to do is take that enzyme inhibitor complex and put it in a dialysis membrane in a large amount of buffer, and that dialysis membrane will allow the small molecules to get out of that membrane. And so it will dialyze out, and you only free enzyme, which is much higher in molecular weight, is retained into the dialysis membrane. So you are able to regain the activity of the enzyme upon infinite dilution. So this would be an example of reversible enzyme inhibition. It's reversible again because it's binding by those 4 non-covalent interactions. So 3 types of inhibitions are known. Competitive inhibition. A competitive inhibition they're showing you enzyme substrate binding to Michaelis complex going to E plus P. If you have a competitive inhibitor, typically inhibitor structurally looks very similar to the substrate, okay, but it's not a substrate. If it's substrate, it will get converted to product. You'll get catalysis. You want to design your inhibitor so that it's not a substrate, but it looks much like substrate. And because it looks much like substrates, it goes and competes for the same active site pocket that substrate competes for. Okay, so they both go and bind to the same active site. And therefore, it is a competitive inhibition. How does it bind? The equilibria is shown here. E plus I binds to EI, non-physical binding, just like ES. It's forming EI complex. Thermodynamic equilibrium inhibition constant is represented by big Ki value. Again, if big Ki value is a large number, that will suggest weaker affinity of inhibitor to the enzyme. It's a smaller number, it will suggest stronger affinity for the inhibitor to the enzyme. So Ki of 2 micromolar versus 2 nanomolar for an inhibitor. 2 nanomolar binds much more potently than 2 micromolar, 1000-fold. That's what big Ki is. See, there is a theme: big Ki, big Km, big Kd, and so on. So it's all the same. That's why I tell you there is a theme there. It's not difficult to understand this. If you have a competitive inhibitor, you can imagine that if you overwhelm this enzyme with a much higher substrate concentration, right? So you have, let's say, 5 micromolar substrate concentration and you added inhibitor. You saw some inhibition. You measure the enzyme activity initial rate and you saw that rate has come down when there was no, compared to when there was no inhibitor. But if it is a competitive inhibitor and you increase the concentration of substrate by 1000-fold, you will see that you actually are not seeing any inhibition now. Why? Because now, because the substrate directly competes with the same binding pocket of the enzyme, and substrate concentration is so much higher than the enzyme inhibitor concentration, you can overcome inhibition. So higher substrate concentrations can overcome inhibition when the inhibitor is a competitive type. What about second type, uncompetitive inhibition? In uncompetitive inhibition, it turns out that inhibitor only binds to the Michaelis complex, which is to the enzyme-substrate complex where the substrate is already bound to the enzyme. Otherwise, it will not bind. So that's why they are showing you Ki prime. Okay, so Ki prime is the same Ki, but they want to distinguish from Ki. So that's why they have another equilibrium constant, Ki prime, where that equilibrium constant represents the ESI decomposing to ES plus I. On competitive inhibitors, this inhibition requires that one or more substrate molecules are bound, right, to the free enzyme form E before the inhibitor can bind. I is usually not structurally similar to substrate. It is a different binding pocket, right? But it definitely binds to the only ES complex. So once substrate binds to enzyme, binding of substrate opens up the binding site for the inhibitor in this case. Okay. So in this case, if you have higher substrate concentration, you cannot overcome inhibition, right? Why? Because if you have higher substrate concentration, you will form more and more of ES. And if you have more and more of ES, you'll form more and more of ESI. And therefore, you will not be overcome inhibition. Higher substrate concentration will not be able to overcome an uncompetitive mechanism of inhibition. What about when you have mixed inhibition, or what we call non-competitive, which are rarely seen? When you have non-competitive inhibitions, your inhibitor binds to both free enzyme form E and the enzyme substrate complex. Okay, so you have both Ki and Ki prime equilibria, right? And so that is what we call mixed or non-competitive inhibition. It is rare, but actually, is seen in certain allosteric enzymes. Okay, so that's the 3 modes of reversible enzyme inhibition. So What happens to the Michaelis-Menten parameters? It turns out that Michaelis-Menten parameters change depending on what type of inhibition we see. You know, what I'm trying to do here is trying to get rid of any mathematics here just for your understanding, so you actually appreciate the concept more so than mathematics. Okay, when you don't have any inhibitor, your apparent Vmax and apparent Km is just the Vmax and Km. When you have competitive inhibitors, your Vmax doesn't change, but your Km does change by a factor of alpha. What is alpha? Alpha is a new parameters that is defined as 1 plus concentration of I by big Ki. That's what alpha is. What is alpha prime? Alpha prime is defined as 1 plus I by big Ki prime. That's a mathematics. But the key is to understand here that when you have a competitive inhibitor, your Vmax doesn't change, but Km does change by a parameter alpha, and alpha is related to the inhibitor concentration and inhibition constant by this equation. When you have uncompetitive inhibition, both Vmax and Km is affected, and they are affected by the same amount, 1 by alpha prime. That's uncompetitive inhibition. This is all you need to know for this class and even beyond, I can tell you. When you have mixed inhibition, your Vmax, apparent Vmax also changes and apparent Km also changes, but they change by different amount. Okay, so Vmax changes by 1 by alpha prime, and Km changes by alpha prime. Again, those relationships are here. So, is there any way to predict, do experiments and figure out if we have a competitive inhibitor, if we have an uncompetitive inhibitor, or if we have a mixed inhibitor? And the answer is yes, using the double reciprocal plot in presence of inhibitor. How do you do this? Very simple. What you do, pay attention to what I'm saying. Okay, you know how this plot was generated, right? All you did was you measured initial rates at different substrate concentrations. You took the reciprocal of them and you plotted, and you try to get your Km and Vmax. You do the same experiment, but in presence of when there is no inhibitor present and when there is different amount of inhibitor present, different concentration of inhibitor present. So if you did that experiment, right, you will see the plot that is shown here on the extreme left. When there is no inhibitor present, this is the one, alpha is 1. That is when no inhibitor present. Remember, when alpha is 1, 1 by V max will be Km by V max, 1 by S, 1 by V max. There is no inhibitor present. That's why alpha is equal to 1 here. That's the bottom line here. Now, if you increase the concentration of I, when alpha is 2 and alpha is 3, you will see that your curves look like this. I going up, you have higher and higher slope, you can see. But if you have a competitive inhibitor, you will see that all of those lines, when you try to plot, you'll see that they all merge at one place on the 1 by V axis. If that's what you see, you know that you have a competitive inhibitor. Why? Because it turns out that Vmax remains the constant, whether you have the inhibitor or you don't have the inhibitor. But your slope changes. Slope changes, and that is what happens in the case of competitive inhibitor. Slope changes by a factor of alpha. That's it. What happens when you have one uncompetitive inhibitor? You do exactly the same experiment, but you will notice that in these cases, your lines don't merge. They don't merge at any place. They are parallel lines. And when you see the parallel lines, you are seeing that your Vmax and alpha, they both change but they all change by the same parameters, right? As you see here. Sorry, yeah, they both change by 1 by alpha prime in case of uncompetitive, right? So again, alpha is 1, there is no inhibitor and increasing constant. When you see such parallel lines, you know that you have an uncompetitive inhibitor that only binds to the enzyme substrate complex. When substrate binds, it creates the binding site for the inhibitor. When you have a mixed inhibitor as shown here in the case, you will often notice that lines will converge, but they will not converge nicely on the 1 by y axis, but they will converge somewhere in the second quadrant in the middle. And if that's the case, you know that your Vmax is changed and Km is changed, Vmax and Km is changed, but by different amounts. And this is a mixed inhibitor because you can see where they are converging. So this is one way to experimentally determine what type of reversible inhibitor you have developed, you have synthesized in the lab if you have tried to do that. And this is a very simple exercise, very easy to do. We do this all the time. And with that, I will stop this lecture. You have a homework that you need to go and see. There is a lot to learn here. You want to write down the Michaelis-Menten equation. You want to pay attention. You want to solve some problems, numerical problems based on Michaelis-Menten equations. And with that, I will stop this lecture right now.